In this procedure, enzyme mixtures for converting lignocellulosic biomass to free sugars are optimized using an automated system known as a GLBRC enzyme platform, or GENPLAT. First, a mixture model is designed in which 4 to 18 distinct enzymes are combined in up to 96 different ratios using design of experiment software. The biomass and enzymes are then pipetted into 96 deep well plates using a liquid handling robotic system. The plates are sealed and incubated for 48 hours at 50 degrees Celsius. Following the incubation, the wells are sampled using an automated liquid handling system, and the free glucose and xylose are measured using a colorimetric enzyme-coupled assay. The design of experiment software is then used to determine which combinations of enzymes yield the maximum free glucose and xylose.
The overall goal of this procedure is to simultaneously test the efficiency of various enzyme cocktails for converting lignocellulosic biomass materials into fermentable sugars for ethanol, with the overall aim of lowering biofuel production costs. Although we use this method mainly for analyzing enzyme cocktails on alkaline pretreated corn stover, it is adaptable to any pretreatment in any feedstock, such as acid-treated poplar, switchgrass, and dried distiller's grains.
This technique can be used to discover novel enzymes that are important for biomass deconstruction and to find better examples of the known enzymes.
Although the enzymes used for GENPLAT may come from various sources, the enzymes used in this demonstration are produced by expressing P-Pix or P-Pix alpha in Pichia pastoris. Grow Pichia cells in 300 ml batches in baffled 500 ml flasks for 3 days. Then, for the next 4 days, perform methanol induction every 24 hours. After the last day of methanol induction, pour the samples into a tangential flow filtration system with a 10 kilodalton cut-off membrane to concentrate and desalt the culture filtrates. Then, perform a buffer exchange with 5 volumes of 25 millimolar sodium acetate, pH 5, and concentrate the sample to bring the volume to between 10 and 25 milliliters. Measure protein using the BCA method with BSA as the standard. The final concentration of the enzyme stock solutions should be 0.5 to 10 mg per milliliter. Store the enzymes in small aliquots in 20% glycerol at -80 degrees Celsius. To create simplex lattice designs, begin by opening the Design Expert program on a PC. Input the number of enzymes to be tested and the upper and lower proportions. For example, a lower proportion of 5% for core enzymes. Then, indicate whether the experimental design is quadratic or cubic. Quadratic is necessary for mixtures with more than 8 components. In Excel, calculate the amount in micrograms of each enzyme to be added to load a total of 15 mg per gram of glucan per sample. Then, calculate the amount of each enzyme in microliters to add to each reaction based on the protein concentrations of the stocks. Generate additional Excel worksheets specifying source labware, source wells, destination labware, destination wells, and transfer volumes for dispensing of water and enzymes according to the experimental design. Finally, import the worksheet into BiomechFX software using the Transfer from File function. To generate a pre-treated corn stover biomass slurry, pour the dry biomass into a Wiley mill and grind it to pass a 0.5 mm screen. Collect the ground biomass in a beaker. The glucan content is measured by standard methods as described in the Jove article, Comprehensive Compositional Analysis of Plant Cell Walls, Part 1, Lignin. Once the glucan content has been determined, mix the biomass with sodium citrate buffer so that the final glucan concentration is 0.5%. Turn on the paddle reservoir and pour the biomass slurry into it. Now, thaw the enzymes on ice. The amount of each enzyme needed is dependent on the size of the experiment. To dispense biomass slurries, generate wide bore tips by using a razor blade to cut the last 0.5 cm off the span 8 100 microliter pipette tips. The trimmed span 8 tips can be reused. Once trimmed, place the tips in a pipette box so that they can be automatically picked up by the span 8. This instrument is equipped with a span 8 pod, with a disposable tip function that can dispense individual solutions into individual wells, and a fixed AP96 pod, which can transfer 96 identical solutions at one time into a 96 well plate. Next, arrange the deck of the liquid handling system. This consists of one 96 deep well reaction plate, a tip box lid containing water, an enzyme rack containing the enzymes in 1.5 ml Eppendorf tubes, and several full tip boxes in the deck positions as programmed in the instrument setup. Once the deck is ready, check the program for any errors. Start the Beckman FX. The span 8 arm will first pipette 200 microliters of the biomass slurry into each well of the 96 well deep well reaction plates. The span 8 pod will then dispense enzymes into each of the wells according to the spreadsheet imported into the Biomech FX software. Once the pipetting is complete, remove the 96 deep well plate from the deck. Seal the plate with a pierceable cap mat, then invert the plate and tap it once or twice to break the surface tension. Place the plate in the hybridization oven for 48 hours. Following the incubation, centrifuge the 96 deep well plate for 3 minutes at 1500 times G in a swinging bucket centrifuge. While the plate is spinning, program the Beckman FX to use the AP96 pod to pipette 100 microliters from each well into a regular 96 well plate. Then, after the spin, place the plate in the instrument and start the pipetting. When the pipetting has finished, incubate the plates at 100 degrees Celsius for 10 minutes on a plate heater to inactivate the enzymes. Centrifuge the plate at 1500 times G for 30 seconds. For GLC measurements, first add 192 microliters of the glucose oxidase peroxidase reagent using a program written for the AP96 pod of the Beckman FX. Then use the AP96 pod to transfer 12 microliters of the supernatant into a regular 96 well plate. Incubate the plates at 50 degrees Celsius for 20 minutes and measure the absorbance at 510 nanometers in a microplate reader. For xylose measurements, add the assay reagents according to the kit instructions. Then, use the AP96 pod to transfer 4 microliters of each well into 384 well plates and measure the absorbance at 340 nanometers. Perform data analysis as described in the accompanying document using Design Expert software. Five substrates and 3 pretreatments were compared under similar conditions of grinding, 0.5 mm particle size, enzyme loading, 15 mg per gram of glucan, and hydrolysis conditions, 48 hours at 50 degrees Celsius. As can be seen here, there are striking differences between feedstocks and between pretreatments. Clearly, the enzyme cocktail Accelerase 1000 alone cannot deal with all of the variability seen in pretreatments and feedstocks. This experiment compares AFEX pretreated corn stover, referred to as AFEX-CS, alkaline peroxide pretreated corn stover, AHPCS, and AFEX pretreated dried distillers grains, AFEX-DDG. The numbers along the top of the bars are the glucose yields as a percentage of the total glucose in the biomass sample. Note that the composition of cocktails differs significantly depending on the pretreatment and the biomass substrate. Although the main use of GENPLAT is to make synthetic mixtures of pure enzymes, it can also be used to produce optimized mixtures of commercial enzyme preparations. Panel A shows a ternary diagram of 4 enzymes optimized for release of glucose from AFEX-DDG. Panel B shows the resulting yields using this optimized mixture compared to 3 other enzyme preparations.
We have just demonstrated the use of GENPLAT to assess biomass breakdown using individual pure enzymes. Each enzyme has a specific catalytic activity that contributes to the full breakdown of plant cell walls into individual sugars. However, it is not necessary to know the precise catalytic activity of an enzyme in order to evaluate its importance in biomass deconstruction using GENPLAT.
Knowing which enzymes are important and in what proportions is critical for guiding bioprospecting for new enzymes, for protein engineering, and for development of consolidated bioprocessing organisms.
Sometimes the results are surprising. For example, we found that for release of glucose from corn stover, a combination of 2 unrelated xylenases is superior to either xylenase alone. We also found that beta-manganase is important for release of glucose from dried distiller's grain, even though this material has a low beta-mannan content.
This method will pave the way for researchers to develop even better enzyme cocktails for a variety of conditions. You don't even have to know what a particular protein does enzymatically in order to test its contribution to lignocellulose deconstruction.
Take into consideration. It is widely recognized that reducing the cost of enzymes is important to the development of an economical lignocellulosic ethanol industry. Currently available commercial enzyme cocktails are complex and poorly defined mixtures of many proteins, and they are adapted mainly for use on acid-pretreated corn stover. In order to accelerate the development of better enzyme cocktails, several laboratories have developed high-throughput platforms for enzyme discovery and characterization. Thank you for watching. Subscribe if it is useful.